ऑडियो प्रोडक्टली बेटे थोड़ा आपको ऑडियो अच्छी आनी चाहिए इसलिए मैंने बेटे The audio is clear? Tell me. Hello Adesh, Akanksha.
Clear,
Yes.
sir.
Okay. So audio will be clear because I have closed this window. So today we will going to study about plant introduction, selection and harvest. So what is plant introduction? If you are suppose you are living in the Nepal, right, and you don't have any new variety of any variety of banana, right? Then what you will do? What you will do if this banana variety does not exist in Nepal? Then first duty is to you will procure it from any other place, right? Am I correct or not? Okay.
Yes, sir.
And this is called as the plant introduction. Okay, this is called because earlier there was no plant, but you have transferred this plant, you have procured this plant from other place, right? Now this because this plant is called as the germplasm, right? This is called as the germplasm. So you are now having on your land genotype of crop plants. So if you are cultivating a plant that is not existing at your own place and now you are procuring it from other place and then you are growing, this is called as the plant introduction, right? Plant introduction. So what is the type of plant introduction? Based on the adaptation, there are two types of plant introduction: primary introduction, secondary introduction, right? So primary introduction variety is well adapted to the new environment, right? Suppose you are procuring banana plant and now you are not facing any challenge in growing. It's a growing, means it's adapted to the new environment. Sometimes it happens the introduced variety is not going to survive. Okay, so you have to cultivate again and again. So this is called as the secondary introduction. Introduced variety may be subjected to selection and hybridization to isolate the superior variety. So you have to practice it again and again, then you are getting the superior variety and then you are growing. So based on the adaptation, two types: primary introduction and secondary introduction. And based on utilization, there is a direct introduction. New variety takes no time for establishment. Indirect introduction, new variety taking much time for the establishment. And exotic variety, foreign variety directly recommended for the commercial cultivation in new environment. What is the exotic variety? Now people are doing with the business with the exotic variety. Means this is the foreign variety, but scientists are recommending directly that you can go with the commercial cultivation in the new environment. Okay? What is the plant introduction agencies? These are the plant introduction agencies in the India: NBPGR, FRI, Forest Research Institute, B.Sc, right? NBPGR, National Bureau of Plant Genomic Research. Okay. So Bureau of Scientific Investigation, B.Sc, worldwide, IBPGR, IBPGRI, and CGIAR. Okay. So these are the two worldwide agencies that can give you the plant. You can procure the plant, you can purchase the plant. And in India, there are three agencies which are responsible for procuring all kind of plants, and you can order them, you can procure any plants from them. So what is the activities of NBPGR? This can be asked in examination. What is the function of NBPGR? What is the purpose of NBPGR? What is the function of NBPGR? National Bureau of
Plant Genetic Research.
National Bureau of Plant Genetic Research. National Bureau of Plant Genetic Research. What is the function? Function is to introduce, supplement of required germplasm. Introduce, supplement of required germplasm. Exploration and collection of valuable germplasm. Exploration, collection of valuable germplasm. Inspection and quarantine. Inspection and quarantine. Testing, multiplication and maintenance of germ plant. Testing, multiplication and maintenance of germ plant. So what is the inspection and quarantine? Quarantine means isolation. In the corona time, everyone has heard about the quarantine. So I think everyone is clear about that quarantine. Quarantine means isolation. Okay. If any plant is infected, you have to quarantine. You have to isolate it so that infection should not go ahead. Okay. Infection should not go ahead. So publishing its exchange and collection list. Setting up natural gene sanctuaries. Okay. So introduce supplement of required germ plasma, exploration and collection of living germ plasma, content, testing, multiplication, maintenance of germ plasma, publishing its exchange and collection list. So what is the source of plant introduction? Maybe center of diversity 1, gene bank 2, gene sanctuaries 3, seed companies 4, and farmers field 5. How many? Can you count them? Tell me, फटाफट, without delay.
सर वो थर्ड वाला समझ में नहीं आया, वो क्या होता है जिन सेंचुरी?
I am explaining. First tell me what is the number. Tell me all the 5. Then I will explain. Okay. First one is center of diversity. What is center of diversity? Means like NBPGR. NBPGR, National Bureau of Plant Genomic Research. They are collecting variety of plants, right? So this is also called the center of diversity. NBPGR is also called a gene bank because every kind of plants germ plasm is there. Bank means money, right? But gene bank, collection of genes. Okay, gene sanctuaries. NBPGR is also type of gene sanctuaries because sanctuaries means just wildlife. Every type of animal you can see in the sanctuaries. Okay, have you gone in the sanctuaries where every type of animals are there? That is called the sanctuary. What is the gene sanctuary? Means every type of plants existing there. What is the seed company? NBPGR is not a seed company and not a farmer's field. Okay. So now you can see in nearby place many seed companies are selling their seeds via small shopkeepers. Okay. So farmers field. Always farmers also have variety of seeds in their store for the future use. Okay. So these are the 5 sources you can get the plant. Center of diversity like NBPGR, FRI, gene bank like NBPGR. It's a gene bank because everything is there, gene sanctuaries, it may be wild, it may be cultivated plants, everything is there, then it's called as gene sanctuary. I think you got your answer. What gene sanctuaries?
Yes, sir.
Sarva?
I got it, sir.
Okay. Any other question? If I'm asking any question, just reply. How many types? Count. Center of diversity first. Gene bank. Second. Third one is Abdesh, Abhishek, Amit, Ankush. Third one is what?
Gene sanctuaries, sir.
Very good. Gene sanctuary. Fourth one, seed companies. Fifth one, farmers field. These are the 5 sources you can procure the plant. That's a source of plant introduction. What is the procedure of plant introduction? Procurement of germ plasm, right? Procurement of germ plasm. What you will do first? You will procure. Means you are going to the Delhi where NBPGR is there. Then you are procuring. You are buying. You are purchasing the germ. Quarantine means directly you are not going to plant in the field. First, what you will do? You will plant them in the isolated field, right? Isolated field. Why you are introducing them into the quarantine? Tell me the reason.
Kind of disease and pest.
Because if they are diseased, because you don't know if they are diseased or healthy. If they are having any disease, right, they can be isolated. Cataloging means Cataloging means what? Suppose same variety of plant you are procuring from different places, right? So you can give them a number, right? From Delhi, this D1, from Simla, this is S1, right? So this is called as the cataloging, indexing, okay? Jump of evaluation. Now, out of the 3 or 4 plant, if S1 is growing well, then only you are going to introduce them into the field. This is called as the job class evaluation. What is the multiplication distribution? And if this is adopting well, this is growing well, this field is better, then this is called as the multiplication and distribution. Correct? Any question from this slide? Procurement of jump class, then quarantine, then cataloging or job class evaluation, and fourth is multiplication and distribution. So there is a next method of procurement of jump class. Suppose you are in the Punjab, and now you are going to Nepal, and if anyone is gifting you, okay, so this is also a kind of procurement, right? Right or not? Or you can directly purchase from the Punjab, right? Or you can collect via moving any place and you find that plant, making the collection. So this is the 3 options: as a gift, as a purchase, as a collection. But you can also store propagules, right? Stem cutting, or sometimes root cuttings. All these are the propagules. A restriction on export and introduction of plant material. Sometimes what happens? What is export? Import means carrying the plant to our country. Export means sending the plant to other countries. Okay. So this is also procurement of jump class. If supply is outside supply is more, then I think it should be stopped. Because in our country this is lacking, so that's why we want to introduce the plant material in different places of our country. So immediately the export should be stopped of that plant. Okay, clear? What is the quarantine and where you can keep the quarantine? Quarantine means keeping materials in isolation. Keeping materials in isolation prevent the spread of disease. Prevent the spread of. Second is fumigation or other treatment. Fumigation or other treatment. According to Destructive Insect and Pest Act 1914, what is the quarantine? Quarantine for different group of plant produced is carried out by different agency. Not only you can keep the plant material in isolation, rather than different agency also are responsible for quarantine. For example, quarantine at seaports, quarantine at airports, quarantine at entry point of land routes. Okay, so these are the places where if they are before the cultivation of the plants, they are kept in quarantine and fumigation is done. Why fumigation is done? What is the role of fumigation? Fumigation means treatment, a kind of treatment if any chance of infection is there. By fumigation you can neutralize it. Right? By fumigation you can neutralize it. What is the cataloging? Cataloging means indexing. Accession number is given. Classification. Accession number is given. Means the name will be same from where you procured and where you are going to plant. This is called the accession. Means if you are searching in the internet also, by same accession number you can have this, all the details. Right? What is the germ class? What is this plant? To forget the name. What the yield? What is the sowing time? Every information is available in the accession. So this gives information regarding name of the species, variety, and place of origin, right? And also various other features are also given. So cataloging of germ plasm collection is published by one agency only, and that agency is called as Gene Bank. Gene Bank. Who is publishing this information? Cataloging Gene Bank. Okay, cataloging of germ plasm is published by Gene Bank. Accession number is information regarding species, variety, place of origin, adaptation. थोड़ा थोड़ा नोट भी करते रहा करो. Right? So introduced material are three types, and they are given the prefix. This is a exotic collection, EC. Indigenous collection, IC. Indigenous wild, ID. Indigenous wild, ID. Purpose of plant reduction, why we are introducing the plant? To obtain new crop variety, right? To obtain new crop variety, for example, Sonara 64, Lerma Rosso wheat, Tier 1, IR8 rice. Okay? So these are the newly introduced plant varieties. These are the new varieties, right, in our country, India. Sonara 64, Lerma Rosso wheat, these are the two wheat varieties. And rice variety, IR8 and Tier. They adopted well, they are going well, and they are giving very good results. Why we are introducing these plants? Because earlier yield was very low. So for the crop improvement, we are introducing new plants. Crop or crop. Also, earlier crop were infected by various disease and pests. Now new variety is not infected by disease and pest. That's why save the crop from disease and pests. For example, coffee was introduced in South America and Africa to prevent losses from leaf rust. To be used in scientific studies. To be used in scientific studies. To be used for aesthetic value. Used for aesthetic value. The important achievement, crops are cultivated extensively after their introduction as new crop species. Induction have been directly released as varieties. Varieties selected from introduction helps to develop varieties through harvest. Induction helps to develop varieties through harvest. Okay? You can develop also variety of new plants by the technique of harvest. Right? Varieties you can select also from introduction. And introduction have been directly released as a variety. Okay? So this is an important achievement. Plants are cultivated extensively after the introduction of new crop species. They are going to be directly released as variety. Variety selected from the introduction have to develop new variety through harvest. What are the merits of plant introduction? The merit of plant production, entirely new crop plant is developed. Entirely new crop plant is developed. And second is quick and economical method of crop. Quick and economical method of crop. Genetic base can be gotten by plant introduction. Production of crops from damage by introduction them into disease-free. Production of crops from damage by introducing them into disease-free. Development of superior varieties through selection, hybridization by using innovation. So you got the merit. entirely new crop you can derive with an economical method of crop protection, crop improvement, and genetic base you can develop, right? New genetic base, protection of crop from damage, disease-free plants, and growing superior variety development also. Yeah. So, these are the merits. How many merits? New crop, you can economical method, genetic base, protection of crop, disease-free plants, development of superior. What is the demerit of plant production? Demerit is sometimes weed can grow well in the new area because of introduced plant material. So now, along with the plant, weed is growing. It's a new problem. You want to grow the plants, but now new problem is there. Number of weeds are growing. So this weed is also difficult to remove. Okay. Introduction of disease along with the new kind of disease to the other crops. Same crop is not infected, but other crops may be infected with the new disease. And introduction of insect pest along with the material. Sometimes new insect pest attack is going on, right? So our old variety is also in the crop along with the new variety. Some plants introduce as ornamental. Some plant introduced as ornamental because of noxious weeds. Plant introduced as ornamental, right? Become noxious weed. So this was earlier realized that if we are introducing new variety of ornamental species, now they become noxious weed. Noxious weed means you cannot kill them, you cannot get rid of them by any kind of stuff. And some introduced plant are threat to ecological. So what is the procedure of plant introduction? Now again you can see, this is a summary. Jump path. First route, individual make a direct request to individual or institution. Second route, individual submit the requirement to NBPCR, right? Or you can collect propagules. So quarantine is necessary, right? First step, quarantine is necessary, and quarantine control is exercised by the NBPCR at prescribed part of country. For example, Mumbai, Calcutta, Madras, right? For at least 3 weeks, so that it could not introduce new disease, it should not introduce new insect pest, it should not be weed. If anything is there, it will be identified in the quarantine within 3 weeks. Cataloging, indexing, right? Accession register. A unique number is given from where this plant was introduced, what is the origin, what is the variety, what is the environmental requirement, what is the temperature, what kind of soil it requires. Okay, the plant material classified in 3 groups. Exotic collection, indigenous collection, and indigenous wild goods. Exotic collection, indigenous collection, indigenous wild goods. Clear? Any question? Any question?
No, sir.
And evaluation is the 4th step, right? If you are introducing the plants, their performance must be evaluated at different institutions of NBPCI or Central Research Institution. Okay? Every state has the Central Research Institution, and they are functioning well for rice, potato, and reduced material are evaluated and maintained by these institutions. Resistant to disease and pest is evaluated and the environment favoring heavy attacks by multiplication distribution of the evaluation. Promising material from production may be increased by multiplication and based on general cultivation and variety of necessary. Most of there are identified by desirable character and intent for production. What is acclimatization? This word can be asked in the examination. Write about the acclimatization, write about the evaluation, write about the quarantine, write about the NBPCR, function of NBPCR. Okay? So note down the homework. The first question is define the term quarantine. Second, define the term acclimatization. Define the term acclimatization. Okay. Define the term acclimatization. Right? Next question is procurement, long question. Procurement of germ plasm. Procurement of germ plasm. And you will also write demerits of plant introduction. This is the homework of today's work. In the evening you will submit. You will upload on the Google Class. Right? Now we will study about the hybridization in crop plants. Are you ready? Hello.
Yes, sir.
Yes, sir. Ready.
नोट कर दिया ना सारा होमवर्क?
Yes, sir.
Hybridization of crop plants. What is the objective and aim of hybridization? Chief objective of hybridization is to create a genetic variation, good variety of genes, good characteristics in the crop plants, right? Because earlier old variety may not be disease free, may not be pest free. So now you are creating that variety by the hybridization technique, so that if any wide variety of plant is having that characteristic of disease free, insect, pest free, right? So that is called as the technique of hybridization. That is the aim of hybridization. Combining breeding, transgressive breeding. So there are various kinds of breeding. What is the combination breeding? Main aim of combination breeding is the transfer of one or more character into single variety. Transfer of one or more character into single variety. Okay. So I have given you a homework because yesterday was a practical, and we discussed a lot about the racimos inside. So go to your nearby area and collect at least 5 plant material of flowers. Okay, 5. Right? Maybe crop variety, maybe ornamental, maybe anything, and classify them. Why this is racimos? Why this is sour? Okay, and submit. That will be counted as a practical number 1. Okay. And
Sir,
second,
we can also use crop flower like tomato.
tomato fruit you should not use, flower you can use. Right? Flower you can use, but I think you can see at least 5 to 6, more than 6, so that you have a firm ground, firm concept of this racimo science. Means oldest at the bottom, new one at the top. And the Simo's central part is the oldest, and the new one is at the bottom. Okay? So this is the opposite, and you have to prove that. This is only say Simo's and this. This will not be other compound, amber, yes, only one. Keval racimo is Simo's and the Clear? Any doubt? So factor number 1 and 2, and that will be evaluated next Monday. And immediately marks will be given. Right? And it will be uploaded online. If anyone left, not completing practical, they will be responsible. Right? So now there will be continuous evaluation. There will be no gap. Every Monday I will take it. So transgressive breeding, combination breeding, transgressive breeding. Transgressive means aim to improve yield. Its contributing character through transgressive segregation means They have to produce the plant F2 generation, F3 generation, right? And you can see that which generation is performing. This is called the transgressive combination breeding. Means you have to see for disease-free plant supports, right? Now you are transferring that characteristic into single variety from other variety. Now you can see the, after growing that variety, that is called the combination breeding. Means in the combination you are inserting the gene, the different varieties, and transgressive breeding is aimed to improve yielding, yield, contributing directly to transgressive service. So this is the biggest answer why we are doing for ablation or plant evolution, because population is increasing day by day and food produce is not sufficient, right? That we have to make balance, otherwise there will be hunger. And also there is a various calamities. We have to preserve also the food materials. Way to enhance agriculture efficiency, cultivate more, right? Achieve higher yield. Restore the productive capacity of already degraded agricultural land, reduce waste and losses in production. Reduce waste and losses. That is also a way, but other technique is achieving higher yield. And also, whatever the waste land is there, make them productive, make them restore their productivity. And also, every land should not be free. Everywhere there should be crop. These all ways you can enhance agriculture. So that's why crop variety improvement, right? And this can be done by the hybridization. Hybridization is the crossing between genetically dissimilar plants. Crossing between, write down the homework. This will be also define hybridization. Define hybridization. Hybridization is the crossing between genetically dissimilar plants to obtain crops having useful and desirable characteristics like disease resistant, good quality, and higher yield. Right? Genetically dissimilar crops useful and desirable characteristics like disease resistant, good quality, and higher. Now, what are the different types of hybridization? This is very important for the examination. Note down. What are the different types of hybridization? For the main examination, it will be chemistry. This question is compulsorily going to gas. And if you are writing here and there, then you are not getting the perfect answer, perfect marks. So there are, based on the taxonomic relationship, you write the complete sentence before classification, right? Based on the taxonomic relationship of two parent, hybridization can be grouped into two types. What is the first one? Tell me. What is the first one?
Intervarietal hybridization.
This is intervarietal hybridization. And second one is distant hybridization. This is very important. Distance means distance. There is no relation between the plants. Distant. Intervarietal means there are different varieties having the different quality, right? So there may be intra-specific, there may be inter-specific. Intra means same, inter means different. Tomato and potato, different. This is inter. Intra means tomato and tomato, potato and potato. This is inter. Right? Intra-generic. Intra word means same species, same genera. Here genera they are same genera. Right? Means Solanaceae family. You can select any plant of Solanaceae family. This is called the intra. Intergeneric means different genera. Right? Different genera. This is called the distant. So what do you mean by the hybridization? A crossing or mating of two plants of dissimilar phenotype. dissimilar phenotype. Why dissimilar phenotype? Why dissimilar phenotype? Because if there will be same genotype, there will be no improvement, no hybridization. So
Because
always,
we are making
because we are making hybrid and there's different genotype, then only you can get the hybrid. Right? So A into B species, the seeds as well the progeny resulting from hybridization are known as hybrid. Progeny of F1 obtained by selfing or intermating of F1 plant and subsequent generation are termed as segregating generation. Segregation. Type of hybridization? Intervarietal and distance. What is intervarietal? Inter means same species. Intervarietal means parents of same species. Right? Simple cross, any species. And if you are crossing the two parents to produce F1 and F1 is self to produce F2 used in the back cross product. You are going to selfing and then back person, then this is called a simple cross. And there is a complex cross. More than two parents are crossed to produce F1 hybrid, which is used to produce F2 and back cross. The cross is known as convergent cross. Convergent cross. Why? Because it brings genes from several parents into single hybrid. Several parents into single hybrid. Three parents are there. Here only simple cross, two parents are there. F1 is there and backward. Here three parents are there. A, B, C. A, B, F1 and F1 with C. Here what we are doing? A, B, F1 and then F1 into F1. No, F1 into F1. Here A, B, F1 and F1 then cross with C. This is called as a complex hybrid because already you are having a hybrid. That is the complex cross. I think this is clear. Intervarietal hybridization means crossing of two varieties of wheat or other crops. Right? Intraspecific means maize. Maize. Gene flow between genetically distinct populations. Natural hybrid generally show intermediate phenotype, artificial hybrid may show transmission phenotype, maize. Interspecific hybridization gene flow between diverse species. Increased heterozygosity can generate two polymorphism. Hybrid may show intermediate, transgressive, and novel type. Novel. This is clear. Distant hybridization means when two species of same genus is crossed. It is known as interspecific hybridization. Variety sativa, variety indica, with Coryza parensis, CO31 rice. When two species of different gender are crossed, it is known as intergenetic hybridization. When two species of different gender are crossed, it is known as inter. For example, triticum with Scale cereal, and this was developed new wheat variety called as the triticum. So now we are going to the poles, right? And then we will start. Poles are visible to you. So main feature of interspecific or intergenetic hybridization. Here.
सर एक्सक्यूज मी सर सर होमवर्क का सर क्वेश्चन क्या था सर वो मुझे मालूम ही नहीं पड़ा सर मैं थोड़ा बिजी था
अब मैं बिजी हूँ ना
सर
आई एम टेकिंग द क्लास नाउ डोंट डिस्टर्ब
अब this म पठाइन्छु कि यतापट्टि ग्रुपमा म पठाइन्छु
ओके सर नो प्रॉब्लम सर
watch the recording
ओके
it is used when the desirable character is not found within the species of a crop, then only you can go for the hybridization. And when it is effective method of transferring desirable gene into cultivated plant from their related cultivated or wild varieties. So it is beneficial only when you are taking the genes, you are hybridizing with that gene which are not present in your present crop rather than present in the other crops of white species. Okay. So if you can see vegetatively in the vegetatively propagated plant like sugarcane and potato, right? This is more beneficial rather than the seed propagated species, more successful. The sugarcane and potato, because this is a vegetatively propagated species. It gives rise to three types of grasses: fully fertile, partially fertile, fully sterile, indifferent crop. It gives rise to three types of grasses: fully fertile, partially fertile, fully sterile, indifferent crop. Fifth, it leads to introgression, which is a transfer of some gene of one species into genome of another. Introgression means transfer of some gene from one species into genome of another. This is called the introgression. Introgression karoge, then it will be good. It will be the improved variety. If one hybrid, two genes are always sterile. Okay, two genes are always sterile. So what you have to do? Fertility has to be restored by doubling the chromosome number through pulse. This is very important. This can be asked. Why? How you can restore the fertility of a F1 hybrids between two genes? Okay, by use of pulse. The selection of pure breeding plants. What are the steps? First one, discussion study, emasculation, then bagging, then seed setting, collection of seed, raising of F1 generation, F1 self-pollinated, seed setting of different plants, collection of some plants, raising F2 generation plant, raising F3 plant, then F4, F5, F6. Okay. So this is the different kind of generation, and then you can see which one is performing better. Okay. So you can select from that. So that is emasculation, bagging. Why it is done for the hardness. Breeding, right? Then collection of seed, and then raising the F1 generation. F1 generation may be sterile, may be fertile. You have to check. You can plant self-pollinated. You will go for the self-pollinated. Then you see the seed characteristics, right? And then you can raise the F2 generation. F3 years. In self-pollinated, there are two things involved in syllabus: self-pollinated crops and cross-pollinated crops. Right? So this can be asked. What are the method of hybridization in self-pollinated crops? What are the methods of? Write on the question. Very important. What are the method of hybridization in self-pollinated crops? The method of hybridization in self-pollinated plants. Method of hybridization in self-pollinated. Pedigree method, bulk method, backcross. Pedigree method, bulk method, backcross. Hybridization in self-pollinated. Right? Process hybridization, emasculation, removal of anther from bisexual flower before the setting of their pollen properties. Right? So emasculation is done for the preventing the cell fertilization. Right? So now you have only female part because you have removed the male part. Now you can use that female part for the different pollen grains. The hybridization with the different pollination with the different pollen grains. Right? So emasculation is not necessary for monoecious. Why? Because there are no chances of any cross. Okay. What is the bagging? After emasculation, flower buds are kept closed in bag made of cloth, plastic or polythene. It is done to prevent pollution through unknown pollen. Okay? So female part is kept covered with plastic or polythene. Pollination through pollen tagging. Female parents are then labeling properly, labeling. So the serial number, detail of male parents or female parents, and date of emasculation. This is the first step. Once if you learn emasculation, you learn the hardness. If you learn bagging, you learn hardness. If you can identify bisexual flower, unisexual flower, then you learn hardness. Okay? Because this is a topic self-pollinated flower. Means already we are talking about the bisexual parents. So there you need a emasculation. There you need to remove the iron because female is the source of progeny formation. Okay, so if you want to improve the variety, use the different pollen. For using the different pollen, you must use the bagging technique. Means flower butter get enclosed in the bags made of flow, plastic, or polythene. Okay, so that is the benefit. Now that pollen you have removed because again that pollen, again the same kind of seed. If you are using a different flower pollen, again you are getting the different pollen. So tagging must be done so that you should not forget. If you are getting the superior variety, then you forget from where anther was there, right? So you should mention everything, date, name of the male parents, female parents. Now graphically you can see this is the emasculation, right? We are removing the male part, anther. Now you can see the second part is bagging. Means after removal of the male part, anther, now we are protecting the female part, that is called bagging and tagging. At the time of harvest. 10:30 बजे तक क्लास है, कितने बजे क्लास है? For the time, really. No. So bagging and tagging at the time of harvest. Collect seed and plant, collect best gene type, known as a commercial cut. This is the step. Male parent from different varieties and female parent, you have transfer pollen grain to stigma, and then you can collect the seed will form, and then select best genotype, you can go and check their performance. So I think today there is a big achievement. What we have learned best, how to improve the variety. You can practice in your field. If you want to grow larger size of chili, for example, that is then you can select the anther from larger variety of chili. And then bagging with your variety of chili. Then you can see the improvement. So now what you can use by the hybridization? Very good resistant, high resistant, yielding capacity depend on from where you are collecting the pollen. High fertility pollen, high fertility seeds, lifelongness, for example, brinjal, ladyfinger, chili, cardamom. So, and there is a pedigree method. Pedigree method is also for selection and segregation observation of self-pollinated plants. There is a two method: hybridization, emasculation, bagging, tagging. Right? In the self-confidence crop, second method is pedigree method. What is the pedigree method? Pedigree method depends on selection. This is also depends on the basis of selection. Early generation test. Right? You have to select the superior process and plant from within superior process. So this is called the pedigree method. Means what? You are selecting the suitable plant for specific characteristics, such as suppose out of 100 plants, one is showing disease, select plant type, better plant type, select better maturity time, select them, right? This is the basis of selection. Pedigree means you are not doing hybridization, you are already selecting the superior process. Okay? You have the better cross, then now you have to select the progeny. This is part of the pedigree. What is the merit, demerit? Take less time than bulk method, improvement of characteristic, provide maximum opportunity for breeder, right? Pedigree record help breeder to obtain population of merchants or quality. Demerits, maintenance of accurate pedigree record, take time, selection and within large number of progeny in every generation, liberation, but liberation time consumed, right? Success of this method legally depends upon this. So it's not in in care is not taken to retain sufficient amount of protein, the living genotype may be lost. What is the bulk breeding? It was also known as the mass selection. Mass, okay? Bulk breeding also called as mass method. Mass or population. Okay? It was first used by Nelson. First used by Nelson arrangement. Okay? A disease is grown in bulk plot type with or without selection of part of pulse is used to the next generation and individual plant selection of pesticides. Means in bulk selection is done. Okay? You are going in the bulk plot from F1 to F5 with or without selection. A part of bulk seed is used to do the next generation and individual plant selection is practiced in later generation. Means पहले नहीं करते. So obviously transgressive segregation can be the result of form selection. Discuss artificial pollination. Artificial pollination. This is the controlled pollination technique. Right? This is the controlled pollination technique to improve the growth seed and other crops. Artificial pollination often have the genetic incompatibility. Male sterility that we call genetically induced prevention of fertilization. Sometimes fertilization is not possible, even if you are carrying the best anther and best stigma. Okay, so there is a prevention of fertilization from other species because some species have a such kind of mechanism. Variety, various techniques are available for hybridization is one technique we have discussed about, right? And white process already discussed. Embryo culture is another very good technique because of infertility issue. Embryo resulting from white process do not develop normally and need to be extracted. This is called the rescue. Means they are not able to grow, they are infertile. Right? Then you have to extract the germ plasm and then go into the plant tissue culture. This is called as the rescue method. By plant tissue culture method, you can grow them into the complete plant. And this is very successful for some genus. Right? For example, cactus. Embryo culture is very useful for cactus. Chromosome doubling already discussed that sometimes F1 hybrids are not fertile. So, colchicine treatment, doubling the chromosome by treating with colchicine. Colchicine used for chromosome doubling. They can improve and restore the double haploid sometimes is working for the removing the infertility. Means double haploid type. When haploid cell fall in array then an event that can produce indigo or in vitro and be spontaneous or artificially induced plant. Haploid may be generated by a wide crossing and then you can do for the doubling of the chromosome. Okay. This is called the artificially induced spontaneous doubling method. So this one practice in barley. Haploid you can generate by wide crossing. So doubling is achieved through colchicine. Right? Double haploid can be cultivar in development in some elite variety. Okay, for example, maize, barley. Okay. So other technique is quantitative trade loci and bulk segment analysis. That can also give you the better results. Sometimes you have to select the genetic marker tool that you can visualize. That is called as the genetic marker. Means you have now to go into the laboratory for finding out the gene and all that. If we have the good genes, you can visualize with your own eyes. This is called as the genetic marker. There are certain genetic markers that is called as the marker assisted selection. That is also called as the technique is also called as the marker assisted selection. MAS. Write down the homework. Next homework is MAS. What is? Define marker assisted selection. Define marker assisted selection. Right? Write down marker assisted selection. MAS is not a genetic engineering method. Rather than you have to select the alien variety of genes, alien genes that is not already present in your class. So your marker may be DNA based, marker may be enzyme based, right? And this technology can help you in identifying if a good character is present in your new problem. Right? This is better than your conventional count. Right? What are the name of marker? You can note down the name of marker: RFLP, SSR, PCR, RAPD, SNP. How many? RFLP, SSR, PCR, RAPD, SNP. RFLP, SSR, PCR, RAPD, SNP. Okay? RFLP means restriction length polymorphism, means satellite, PCR based marker, RAPD, and single nucleotide compound. Single nucleotide polymorphism. What is quantitative trait? Number of gene. Controlling a quantitative trait. Unfortunately, most traits are interest. So number of genes controlling a quantitative trait, and most traits of interest quantities are controlled by only genes. This contributing a small effect to the overall individual. So 12 quantitative traits are controlled by polygenes. Means they are linked, they are the marker. 12 is also kind of marker. Various genes are there. And multiple, since traits are controlled by the multiple genes, that's why this is called the polygenic traits. Quantitative traits is also called the polygenic. Polygenic inheritance is in the type pattern that is continuous because evolution occurs in large. As a consequence, quantitative traits are more susceptible than qualitative traits. Modification by variation in factor which plant in the population are. Method of plant breeding in self-pollinated selection. These are two types: pure line selection, and mass selection. What do you do in the self-pollinated selection method? Pure line selection and mass selection. What is the pure line selection? Pure line selection has been the most commonly used method of self-pollinated crops. Almost all the present day varieties of crops are pure lines. Pure line selection has several applications. Improvement of self-pollinated crops. It is used to improve local varieties, old pure line varieties, and introduce the general procedure for evolving a variety of pure line selection. Selection of individual plant from local variety or some other mixed population. This is evaluation of individual plant colonies and yield charge. These are the pure line selection. Means improvement for self-pollinated crops. Right? What you are doing in this technique? You can improve the local variety. You can introduce new variety also. By this technique, you can improve the old variety. You can select the individual plant from its population, and visually you can see which individual plant is best. So, in the various generation, you can see the input, not one generation. This is very level. This is a symmetric generation of pure line selection itself. First year. Now a mixture of pure lines you have. Second year, now you are segregating the better individual plants. Third year, now you are selecting those plants from the second year which are performing well. Fourth to sixth year, Multi-location in trial, 7th, seed multiplication. Okay. Right? This is performing various genes. What is the advantage of pure selection? Pure selection are extremely uniform, attractive and liked by the farmers and consumers, cool and stable. And due to extreme uniformity, the variety can be easily identified in the seed surface. Disadvantage of pure line selection: new genotypes are not treated by pure line selection. This method is applicable to self-pollinated crops. Improvement is limited to isolation of the best genotype present in. This is the achievement of rice. You can see there is variety, sorghum, groundnut, red gum, gram, chilies. The procedure for evolving variety by mass selection. Large number of phenotypically similar plants having desirable characters are selected. Secondly, composite seed plant in standard checks. Third, to 50 years, the variety is evaluated in coordinated yield trials at several locations. Now we are going to the PROF, the plants, same plants which are performing better at a different location, different state, okay? And if they are giving the same results, then they are good. 6 years, the variety is proved superior in main yield trial as compared to the old varieties. Then it is multiplied and raised up to well, first year, second year, fifth year, and sixth year. You are getting the seed multiplication in the 6th year if they are performing better at the different places. This is called the mass selection in self-pollinated crops. Now write down the next homework, how will you perform mass selection in self-pollinated crops. Okay. So what is the merits? What are the merits? Demerits? Can be practiced both in self and cross-pollinated crops. Mass selection are more widely adopted than pure lines. It retains considerable variability and hence further possible in future by selection, help in preservation of line bases, useful for purification of pure line varieties, improvement of character, genes, less time consuming and less expensive. Demerit of mass selection: variety are not uniform. Since progeny test is not done, so genotype of selected plant is not known. And since selection is based on phenotype, there is no control over pollination, right? So you can get the improved variety or not. This is not sure. Process of mass selection has to be repeated and very laborious. Technique, character which are governed by large number of genes with low heritability cannot be improved. Can create new genotype but utilize existing genetic. These are the results: potato, rice, sorghum, bajra, groundnuts, cotton. Okay. Already discussed open pollinated. What is the open pollinated population cultivars? You note down, this term is often asked to the examination. What is open pollinated population cultivar? And most of the students are helpless at that time when the question is asked. So I think you must know the maximum term and start writing as soon as you get the question. So write down its definition. In open population selection of desirable cultivar is usually carried out by mass selection, recurrent phenotype selection, or selection in progeny testing. The maintenance of these cultivar is through open pollinated population with uncontrolled means random mating. Means open population means random mating is done. Right? Uncontrolled mating. Okay? Selection of desirable characteristic can be done by the 3 techniques: mass selection, recurrent, and phenotypic selection with progeny test. Okay? Mass selection already we have discussed. What is synthetic cultivar? A synthetic cultivar basically gives the same end result in an open cultivar. The main difference that a synthetic cultivar is continuously reproduced on specific parents. Where it is left to open pollinated generation, it will change its genetic makeup as a population. This means the farmers need to return the seed company for a new seed and then they can raise it. This is synthetic cultivar. Right? Breeding method used for the development of synthetic cultivar. Depend on the ability to develop homogeneous line that is present in the parent or restricted to the parents. Propagation by any phenotype. See? What is the recurrent phenotypic selection? Means a recurrent phenotypic selection more effective than mass selection. The population is created by cross pollination between two populations to create. It's called the base population, right? This is created by cross pollination. This is a base population. Large number of plants are grown from base population, right? And then subsample of the most are identified and harvested. Okay. These plants are randomly mated to new improved population. This process is repeated a number of times, and that's why this is called the recurrent process. First, process is repeated many times. That's why recurrent phenotypic means you have to produce cross-pollinated variety population. This is called the base population and select best one. Then go for intercross selection and then improve. And you are repeatedly doing this, that's why you have better chances of improving the population. Bulk method already I have discussed you. In the bulk method, we are creating the integration by hybridization between two parents, the F1 and several subsequent generations. Generation F6, F7, F8, F9 have to go for the different trials. Okay? So major steps in producing hybrids are very similar to the synthetic cultivar. Very similar to cultivar, bulk method. Right? Selection is not imposed, that is the best part, and you have to select out of the best bulk part. So means P1, P2, then F1, then F5, and then you have to select best plant from the F1, individual plant from the F1, grow in the F6, and then F7, then F8, then F9. Better you can say. Right? So it must show a high degree of heterosis, capable of being handled as to produce inexpensive hybrid seed, not easily produced uniformly by other means under a premium or crop uniformly. What is heterosis? This question is asked many times. What is heterosis? Define heterosis. Right? Heterosis means performance of a hybrid is a function of genes. Right? We are selecting a parent that has the better genes and they will perform better. Right? What is the heterosis? Heterosis means better performance. Phenotypically better performance. This is heterosis. Right? Many breeders believe that magnitude of heterosis is directly related to the genetic diversity, degree of genetic diversity. Means if genetic diversity is better, they will show better heterosis, means better hybrid plants, seeds, okay? Better hybrid germplasm. Okay? So I have given some question. Already discussed concept of breeding, self-pollinated, cross-pollinated, and asexually propagated plants. Self-pollinated species, you can see what are the plants: Histogamy, Plasmogamy, bisexuality. They develop self-fertilization, autogamy. Right. So all these features we are ending this lecture now. If you have any question, you can ask.